All right, ladies and gentlemen, welcome back. We're looking at the first section of our DNA unit, and it's all about what the structure is of DNA and how it's replicated. So this first one just shows a little cartoon of how someone's describing DNA. I will expect you to know it in a lot more detail with the actual words. Okay, so here is the structure. It's made up of nucleic acid. It's a polymer of nucleotides. Nucleotides from our biomolecules unit are the basic building blocks of nucleic acid, and it breaks down into three parts: phosphate, the sugar, and then the base. And as you can see here, here is your phosphate group, here is your sugar group, and here is your base. This specific sugar is deoxyribose, and that's where we get the deoxyribonucleic acid from, also known as DNA. The other thing I want you to take note of is that see the 1, 2, 3, 4, 5. This little apostrophe means prime. So when we start talking about 3' and 5', we're essentially referencing the carbon number of this pentose sugar. So this is the first carbon, second, third, fourth, and then the other one's up here connecting up to the phosphate for 5. Okay? So you may see that again, and by may I mean you will. Okay, so back to the nitrogen bases. We have purines, and we also have pyrimidines. Purine just simply refers to having a double ring structure, like adenine here on our right, and pyrimidine is a single ring structure, like thymine and cytosine. And all of these bases are going to be complementary. And so the complementary pairs, let me just get the pen here, sorry about my writing, it's awful, would be adenine and thymine. Those are complementary pairs, and also cytosine, that's cytosine, and guanine. Okay, so you can jot those ones down, and I'll show you why they're complementary here in a second. So we mentioned that we have phosphate and sugar, and what that makes up is essentially what we call a backbone. And what it is it essentially connects from one side to the next by these hydrogen bases between the nitrogen hydrogen bonds, sorry. Okay, as you can see, there's essentially two strands. It's almost mirror images with different nucleotides. And what happens is when you have a large section of this DNA, it makes up something called genes. And the genes are essentially has the code for the order of amino acids to make a protein. So when we think about function of DNA, it's not that it controls everything, but it does. But we'd like to be more detailed. DNA has the code to produce the right sequence of amino acids, which is going to make the right protein. Okay, as you guys can see here too, we have 3' and 5' like I talked about in the last slide, but we actually have 3' and 5' ends. So what it is complementary. One end will start with 5 and the other one will end in 3. And then on the other sides too, they're complementary. So if this is a 3 side, this is the 5. And again, the 3 and the 5 refer to the number of carbons, or the carbon number on the sugar. Okay, so this is something that we'll see again in class if that's at all confusing, because I think it was. Okay, so let's look at the replication process. It all starts with enzymes, and these enzymes are going to be responsible for changing one DNA helix into 2 helices. The reason why we need it is for cell division and growth and repair. In order to have extra cells, we need extra DNA, and in order to have extra DNA, we need to replicate it. And then finally, before we get into the process here, is that you may notice that this DNA helix ends up breaking apart, and so these new helixes will essentially have one strand of old and then the one strand of new. So we call it semi-conservative. So you can pause that to get that down. So let's look at the process of replication here. We have DNA helicase, which is going to unwind, so turn that coil into like the ladder shape, and unzip, essentially breaking the hydrogen bonds between the adjacent nucleotides. Okay, so that's the first step. Break it apart, and now we can do something with it. Again, notice 3', 5'.
Helicase is marvelous, unwinding the double helix at breakneck speed, slicing open those loose hydrogen bonds between the base pairs. The point where the splitting starts is known as the replication fork. It has a top strand called the leading strand, or the good guy strand as I call it, another bottom strand called the lagging strand, which I like to call the scumbag strand, because it is a pain in the butt.
So what's going to happen now is there's these enzyme's called primase, RNA primase, and it's going to lay down an RNA primer. And what RNA essentially is very similar to DNA, but with just some slight differences that we'll go over again in class. So what we have here is we put these primers, these nucleotides, that are going to be essentially the starting spot in order to be able to copy this. So here it is. So the big one, the main guy here, the enzyme is DNA polymerase. And what it does is it adds complementary DNA nucleotides to those existing strands, but it lays it down from 5' to 3'. Okay? So if you look, since you have complementary strands, this one here can lay it from 5' to 3', going right towards where it's being broken apart by helicase, and it's nice and continuous. But the problem here with this one is that this needs to add it from 5' to 3', but every time this part's breaking up, it needs to backtrack and go this direction, and backtrack and go this direction. So that's why those primers are so important, because it lays the primer down. DNA polymerase essentially codes it, or will add the nucleotides. When it gets to the end, it will go back, go to another primer, and start again. Whereas this one only needs the one RNA primer because it doesn't stop. It just goes continuous. So we have something that's called the leading strand, which means it's continuous and not stopping. And then the one that's always lagging behind, so we call it the lagging strand. These little fragments here have a name, and they're called Okazaki fragments. Once this is done, there's going to be some red regions here, which is RNA, but we don't want RNA, we want DNA. And so what the DNA polymerase is going to do when it's done coding, it's going to come back here and replace those primers with DNA.
The enzyme RNA primase. The leading strand only needs this RNA primer once at the very beginning. Then DNA polymerase is all, I got this, and it just follows the unzipping, adding new nucleotides to the chain continuously all the way down the molecule. Copying the lagging or scumbag strand is, well, he's a freaking scumbag. This is because DNA polymerase can only copy strands in the 5', 3' direction, and the lagging strand is 3', 5'. So DNA polymerase can only add new nucleotides to the 3' end of a primer, so maybe the real scumbag here is the DNA polymerase. Since the lagging strand runs in the opposite direction, it has to be copied in a series of segments. And here that awesome little enzyme RNA primase does its thing again, laying down an occasional short little RNA primer that gives the DNA polymerase a starting point to then work backwards along the strand. This is done in a ton of individual segments, each 1,000 to 2,000 base pairs long, each starting with an RNA primer. These are called Okazaki fragments, after the couple of married scientists who discovered this step in the process in the 1960s. And thank goodness they were married so that we could just call them Okazaki fragments instead of Okazaki-someone's-someone fragments. These allow the strands to be synthesized in short bursts, and then another kind of DNA polymerase has to go back over and replace all of those RNA primers, and then the little fragments gets joined up by a final enzyme called DNA ligase.
Okay, so to finish it all off, there are sometimes a few nicks, which basically means that there's going to be a break here. When these nucleotides were added, it wasn't sealed. So phosphate on one and sugar on the other wasn't sealed together. So DNA ligase is what's going to carry out that function. So it fixes any breaks in the phosphate sugar backbone. So you can pause that to get that done. And here's the whole process. Oops. So here's the whole process. Again, so we have our helicase breaking apart the nucleotides in the right direction from 5' to 3'. We have our RNA primer. DNA polymerase will bind to that and start adding the complementary nucleotides. Same thing here. We have our RNA primers that is essentially going to be allowing our DNA polymerase to add to it, but it's got to go in this direction away. So it has to do it in those Okazaki fragments. Okay, so we'll go over this in a tremendous amount of detail next day, and have yourselves a wonderful night.